Hello, everyone. Welcome to the fifth MaxWell webinar. For today, we are broadcasting live from Switzerland and from the US. Thank you very much for joining us. So this is a very special webinar because this is co-hosted together with FUJIFILM Cellular Dynamics, and we also have a guest speaker from ETH Zürich. So let me introduce our co-host and also our guest speaker. So first, our co-host, Simon Hilcove from FUJIFILM Cellular Dynamics. So we're very happy to have them hosting this webinar with us today. And they are basically manufacturing and also providing human iPS-derived neurons, cardiomyocytes, for the use in basic research and also for drug discovery. Simon, say hi to our audience. Thanks, Simon.
Hi, everybody. Great to be here.
Thanks. So we have a joint protocol that we have developed together. We have written together, which basically describes how to plate FCDI cells on our chips, MaxWell and Max2. So you can download this from the chat that you can see here on the right side in a few minutes. Okay, so next we have our guest speaker from ETH Zürich, Silvia Ronchi. She is a doctorate student in the bioengineering laboratory supervised by Professor Andreas Hirtmann. And currently the work that she is introducing in this webinar has been published in bioRxiv, which you'll also see the link here in the chat on the right side. So Silvia, hi.
Hello, everybody.
Thanks, Silvia. Okay. So let's continue. So again, let me show you the files that can be downloaded is here inside the files on the chat. And within the chat itself, you can see a comment on top linking to the bioRxiv paper of Silvia. All right. So let's continue. My name, by the way, is Maria Obien, if you don't know me yet, from Assoc Biosystems. Okay. So for this webinar, we will first introduce the technology side here at MaxWell Biosystems, and then the cells from FCDI side, and then the combination of these two in Silvia's presentation. So for my presentation, it's entitled High Content Electrophysiology Platforms for Characterizing Human iPSC-derived Neurons, and I will introduce these high content electrophysiology platforms. So first about our company, MaxWell Biosystems. We founded the company in 2016, and since then, we have been providing electrophysiology systems and solutions to advance scientific discovery and to accelerate the development of novel drugs for brain diseases. Our expertise here in the company include MEA platforms, both on the technology side and also on the application side. And our customers are academic labs, contract research organizations, biotech, and pharmaceutical companies. You can see here on the left side our new team picture. We are growing, and we are very happy to show this new picture to you. All right. So why are we actually doing this? What is the motivation? First, let me show you this slide wherein we have the pipeline for drug discovery for neurodegenerative diseases. And here you can see that there's a lot of failures going on. And the reason for that is because we have poor clinical prediction of the efficacy and safety of drug candidates in humans. We have been testing these compounds early on in animal models, in vitro, not yet in humans. And especially for the brain, this is quite different from other types of cells. There are very different levels of activity that happens that we need to be able to characterize. So MaxWell Biosystems will basically be involved in the beginning of the pipeline of the drug discovery. That is here in between drug discovery and preclinical phase, focusing on in vitro experiments. And also the green cell here corresponds to human iPS cells. So this is an upside potential because now we are able to access human cells in vitro. With human induced-pluripotent-stem-cell technology, we can have highly reproducible cells which are having patient genetic background that are human, and we can also perform high throughput experiments. And this is much better as compared to animal models. So we can actually have these compounds being able to be tested on human cells earlier. And with this, we also have a need for high-quality readouts to be able to characterize these human iPS cells and also the effects of compounds. And this is where we come in. So our technology is an electrophysiology system. So on one side, patch clamp is the gold standard where we have a micropipette that is placed on individual cells to be able to detect activity of these individual cells. On the other side, we have imaging that allows us to identify how cells form into networks, how they grow, and also how they mature and if they die. Our technology is kind of a combination of these two technologies. It's high-density microelectrode-arrays that detects action potentials from individual cells, but because we have many electrodes, we are able to extract the propagation of such signals from one cell to the next. So this is an example of activity of one cell where it initiates at the axonal initial segment and propagates along the axons, which allows us to also extract the morphology of such a cell electrically without staining the cell. And here on the inset, you see the different activities of different cells. Each spike corresponds to an activity of one cell. So again, what do we measure? Intracellularly for patch clamp, we have these positive spikes corresponding to action potentials. And these are the same things that we can record extracellularly. It's just that we see them as negative spikes from outside of the cell. And with high-density microelectrode arrays, we have multiple of these electrodes that are tightly spaced with each other. So we are not only detecting signals from one point of the cell, but also from multiple points of the cell, such as along the axon, at the soma, and also along the dendrites. So what are the key advantages of high-density microelectrode arrays compared to the low-resolution ones? First, we can see all of the cells. We have electrodes everywhere to be able to identify the activity of each cell. So here we cannot know whether the cell the activity is coming from the green, the red, or the blue. But for high resolution, we have electrodes closer to the neurons who are really able to identify if the signal is coming from the green, the red, or the blue cell. We always have electrode at the right spot, and we also have access to small signals. We don't miss such cells like this because we always have electrodes everywhere. We can also read out at different scales, at the population network level, at the cellular level, and even at subcellular level as what I've shown you earlier. So what are our products? We have MaxWell, which is our single-well microelectrode array, and Max2, which is the multi-well version. And we have 6 and 24 well plates. The main frame for Max2 is also a built-in incubator, so you can do your experiments outside of the incubator. Now, the most important part of this is actually the chip inside of each well. And for every well, we have this sensor, which has 26,400 electrodes within 2 by 4 millimeter square area. And the distance between the two electrodes is 17.5 micrometers center to center. We also have low noise readouts, which is very important to be able to access the low noise signals that are coming from the neurites or the axons that are propagating along these compartments of the cell. And any of the 26,400 electrodes can also be used for electrical stimulation. This means that any of these cells can be activated separately using our technology. Again, another part of our product is also the software. So we call it MaxLab Live. This is an all-in-one software that allows us to access different parts of the data, visualize them, acquire them, organize experiments, and also to generate summaries and also reports from these results. And the software is also able to control the hardware itself, so like opening and closing of Max2 and also controlling the holding of the plate. So this is an example of a live data visualization from one part of the array. And you can see that you can find out where the individual cells are. Whenever you see them shining, that means they are active. And you can also see whenever there's a burst, like now, when all of the cells are active at the same time. So what can we do with our software and with our device? So we have these neuronal assays. Of course, we can also use the technology for other cell types like cardiomyocytes, muscles, and other electrogenic cells. But for this webinar, we're focusing on neuronal assays. We can do activity scan, network, and axon tracking. And going through the three of them in the next slides. So for activity scan, we can ask, are the neurons active? Basically here we are scanning all the 26,400 electrodes to be able to find out where the active cells are. Comparing the microscopy image here, we see where the cells are, which are also the same as what you can see from a MAX2 or MAX1 functional image. And not only do you know where the cells are, you can also find out how active they are. So this is showing the firing rate of the cells per pixel. So this is an area where there are highly active cells, and the light green parts are those with less active cells. Next, we have network connectivity assay. Here we ask, are the neurons interconnected? And we can find that out by checking how burst features are and how synchronous the firing rate of the cells are. So how do we do that? So each of these lines in the raster plot corresponds to the spiking activity of each of the cell, and each dot corresponds to one of these spikes or negative inflection in the signal. So here, whenever you see a vertical bar, that means all of the cells are active at the same time. And that happens when there's already a network being formed in the sample. And here's a summary of the network activity. This is high whenever we have a lot of spikes going on at the same time, and this is low when there are less spikes. And then we have the axon tracking assay. Here we can ask, are the axons functional? And we can extract the solar arbors of single neurons and also calculate the conduction velocity of each of the axons. We can also follow the same cell across multiple days. Here is an example from day 17 to day 30. And this is how we can track the action potential propagation for one branch of a cell. So for Silvia's results later, you can see these results for different cell types, and I'm excited to see these. So stay tuned. Okay, so what are the different neuroscience applications? We have 2D samples, such as from human iPSC-derived neuronal cultures and also primary neuron cultures. We have 3D samples from brain slices, organoids, and also neuronal spheroids. And we also have the retina, which we can combine with light stimulation to be able to find out how the retina reacts to this stimulation. So that's it from MaxWell Biosystems side. I will introduce Simon for cellular dynamics. Simon, you can take over. Yep. Thank you so much.
Okay. Thank you. Wonderful. You can hear me okay?
Yep. Perfect.
Okay. And screen.
Yeah, so maybe just for everyone in the audience, feel free to ask your questions in the chat anytime. We will answer them by the end of the webinar. Okay.
Okay, you can see my slides okay?
Yep.
Okay, perfect. Thanks so much. So I wanted to start with just describing a little bit about FUJIFILM cellular dynamics. So we're based out of Madison, Wisconsin, USA. We're really a good biotech company here that's really built on iPSC technology. So Marie mentioned briefly a little bit about what that is. And in our case, what we do a lot of is, a big part of what we do is taking episomal reprogramming factors, introducing those into peripheral blood or skin samples that are derived from or taken from healthy donors or patient-derived. And then through this process, you're able to induce them into a pluripotent stem cell state. And what's cool about induced pluripotent stem cells, of course, is that they're capable of generating, in theory, any cell type in the body. So this is the other big area that CDI does a lot of focus on is optimizing differentiation. So we're really good at making cardiomyocytes, neurons, as we're going to talk about today, hepatocytes, and some other cell types. Another aspect that we'll talk a little bit about today is another area where CDI does some focus is genetic engineering. So what's another good aspect about iPSCs is that you can use nuclease-mediated engineering techniques to introduce point mutations to study disease or correct disease, and then do really interesting methods of disease modeling in that sense. So So we provide all these cells in kind of cryopreserved, ready to go, making it easy for people to work. And then on top of that, what we do a lot of work on is application support. So just trying to make it easier for people to take these cells and put them into assays that are going to be fruitful in their drug discovery or their basic science research. So CDI does also have a lot of efforts in the cell therapy space, but that's not something we'll be covering today. So we're up to about 15 cell types and counting. One of the big areas we have a lot of focus on is in the nervous system. These are difficult cells to use or there's not a lot of access to human cells from the brain and the heart. So this is a big area that we've focused on providing these. The cells are really widely published. They're kind of becoming almost the gold standard in a lot of ways for doing this kind of work. Because we spend so much time focusing on quality and scale up and consistency. It's one of the best ways to ensure you're going to get the same results. Well to well, or experiment to experiment, or even from lab to lab across the world. If everybody's using the same cell type, that can go a long way to getting that reproducibility. Within our neural portfolio of cells, of course, we have motor neurons, which we're going to talk a little bit about today, our dopaminergic neurons, and one of our newer products, which is not being mentioned today, is microglia. So to look across our portfolio of neural cells, one of the first cell types that we generated was isol GABA neurons, so these are kind of a primarily inhibitory type of neuron. So primarily GABAergic. They're cortical neurons, and they, as sort of a complement to this, we have the isol glutaneurons, which are primarily glutamatergic. They're excitatory. So over time, they'll form synchronous bursting networks. So this is an example of a heat map forming on a regular MEA plate. You can see the cells bursting in synchrony. Each one of those is an electrode. So in this case, there's looks like 16 electrodes per well. And then we also have isol astrocytes, which are a great cell type to include. Of course, they do all kinds of great functions in terms of modulating network activity, and so all three of these are highly pure. Typically 90-95% pure, as you can see by some of the flow diagrams here. Where we've expanded this portfolio more recently was isol dopaminerons. So these are primarily a kind of representing a pure population of human floor plate derived midbrain dopaminergic neurons. They're also highly pure, generally greater than 70% positive for TH, or tyrosine hydroxylase, a classical marker for the dopaminergic neuron. We make motor neurons, which Silvia's going to talk a little bit more about later as well. So these are also highly pure. After plating, they get up to by day 14, when you would want to be doing your assay. They're typically greater than 90% positive for ILIP1. And then our newest product, isol microglia, are again a highly pure population. They express all the markers you'd expect from a human microglia cell, and are capable of a lot of functions of microglia, such as phagocytosis. We have some excellent protocols available for use of these on our website as well. So I mentioned briefly about what methods for doing genetic engineering and disease modeling. So we like to think of the disease modeling in three conceptual ways. So the first one is with innate disease modeling. So you can imagine in this case, you would be taking a healthy donor, reprogramming to iPSC, and then differentiating it to a given cell type, say a motor neuron, for example. And then for a comparison to kind of be linking the genetics to the phenotype, you could do the same thing, but with a patient that perhaps has a disease-causing mutation for, say, ALS. And then when you have you can kind of hopefully recapitulate that disease in a dish phenotype and compare it to the healthy cells. Another way you can do this on the far right is to take just healthy donor cells and then make iPSCs and then use healthy conditions to culture them as a healthy cell, or use disease-inducing conditions to induce the disease phenotype. So examples of this might be using like a high glucose media to look at diabetic cardiomyopathy, or using like adding amyloid beta fragments to induce some neurotoxic effects with neurons. And then finally, what Silvia's going to focus on a little bit is the engineered model. So some of the ones that we've made that Silvia's been able to use are, we've taken healthy donors and generated, as I mentioned, dopaminergic neurons and motor neurons. But what we've also been able to do is take those iPSCs and engineer in disease-causing mutations. So in the case of the dopaminergic neurons, we've engineered in the highly penetrant A53T alpha-synuclein mutation. So it's a single point mutation that causes and is one of the most penetrant mutations. So when you get these two cell types, then what you're able to do is have really the only difference between them being that single base pair, and it's so what we call an isogenic control, and is an excellent way to really isolate that variable and correlate the genotype to the phenotype. And the second one that we've done is generated a couple of TDP43 mutations, which are and made motor neurons from these. And these are TDP43 is, of course, implicated in ALS or motor neuron disease. So overall, we've been making as we mentioned, Parkinson's disease stuff, ALS disease, which Silvia's going to talk about. We do a lot of work in the Alzheimer's space. You can check our website for all kinds of other models we're making in the AD space, as well as some for neurodevelopmental disorders. But with that, happy to turn it over to Silvia. So, thank you.
Thank you. Hope you can hear me. Welcome to my presentation. My name is Silvia Ronchi. I'm a PhD student in the bioengineering laboratory of ETH Zürich, and today the title of my presentation will be the electrophysiological phenotype characterization of human induced pluripotent stem cell derived neuronal cell lines using high-density microelectrode-arrays. The reason why this project started was to offer tools for high-resolution and high-throughput electrophysiology. To do so, we wanted to use some cell lines that could be a reliable model both for drug screening and disease modeling. Moreover, we wanted to study the development of new functional metrics for neurodegenerative diseases. Today, I would like to show you how by combining high-density microelectrode-array and iPSC's technology, we could study the electrical phenotype characterization of different cell lines. We could also discover new electrophysiological disease markers, study the compound effects at single cell and network levels, not just in one day, but over multiple days, and we could also perform recording of the specific differentiation stages. To do this we used the MaxWell MEA that was first developed in my lab, and now it's commercialized by MaxWell Biosystems. To perform the recordings, we used the MaxLab-like software. We performed the recordings once a week from the in-vitro 7 to the in-vitro 42, and we used a total number of 83 MaxWell MEAs. We purchased the cell lines from FUJIFILM Cellular Dynamics. We used 2 lines of human motor neurons, a wild-type line and an isogenic disease line modeling amyotrophic lateral sclerosis. Then we used 2 lines of human dopaminergic neurons, again a wild-type line, and an isogenic disease line modeling Parkinson's disease. And we co-cultured human neurons with human astrocytes in a ratio of 5:1. On the right side of the slide, you can see the MaxWell MEA, the cell culture chamber where we add the medium, and the red rectangle in the middle is the electrode array. In this area here, we plated our neurons, and in particular, we plated 100,000 neurons in this area. Below, you can see a zoom of the array, where neurons were stained and colored in red and astrocytes in green. And on the top right corner, you can see also a schematic of the electrode dimension and also of the spacing between the electrodes. Now, before going through the results section, I would like to show you some of the metrics that we used. The first metrics come from the activity scan assay that Maria already introduced. What you see on top of the slide is a trace recorded by one electrode over a certain period of time. Every time that you see a blue triangle, that's where an action potential is happening. And you see it negative because extracellular action potentials are negative. By counting the number of spikes over a certain time period, it's possible to derive the firing rate. We could also assign a firing rate to each electrode and build up a map as one of the 4 that you see below, in particular the first one, of the area of the microelectrode-array. The dark regions are the one with the lower firing rate, while the light regions are the one with the higher firing rate. Then we could do the same with the spike amplitude, and that's represented in the second map. In this case, the amplitude was represented with its absolute value. We could also compute the interspike interval above represented in green, which is the time difference between consecutive spikes. And in particular, also, we could derive the coefficient of variation of the interspike interval, which gives a measure of the spike regularity. And lastly, we could compute the number of active electrodes over the total number of electrodes. And the active electrodes are the ones that would pass some thresholds, for example, on the firing rate and on the spike amplitude. Now, moving to the network assay metrics, as you may already know, neurons, when are cultured in vitro, after a few days start to form connections between each other. And then they have this synchronous activity that we can call network burst. Maria already showed you a plot similar to this one. From this kind of activity, we could compute the burst duration, which is the time difference between the end point and the start point of each burst. We could also derive the interburst interval, so again, the time difference between starting points of two consecutive bursts. Also the coefficient of variation, which is gives an idea and a measure of the burst regularity, and we could also analyze the burst shape. Now, going to the result, what you see here on top of the slide is one single MEA, where we plated wild-type human motor neurons that we recorded at the in vitro 7, 14, and 21. And you can appreciate how the overall activity, and in this case the firing rate, increased over time. Also looking at the second line, where wild-type human dopaminergic neurons were plated, you can appreciate an increase in activity. However, if you compare the in vitro 21, for example, there is a big difference between the overall activity of the two cell lines. So we decided to compare at first the two wild-type lines. And you can see it in the bottom part of the slide. In these four plots, every point represents one MEA as specific to in vitro. In blue, you can find the results for the human motor neurons, and in red for the human dopaminergic neurons. So we compared these two wild-type lines for the mean firing rate, and we discovered that the human motor neurons were characterized in general by a higher mean firing rate than the human dopaminergic neurons. They were also characterized by a higher mean spike amplitude and a higher percentage of active electrodes. Instead, the interspike interval coefficient of variation was very comparable between the two cell lines, so they had very similar spiking patterns. So using these four parameters, we could find initial differences between the wild-type line. And then we wanted to investigate also differences between the disease line and the healthy lines. So at first, we started with the human motor neurons, the wild-type line and isogenic disease line. What you see on top of the slide is the bursting activity of one wild-type MEA in darker blue and the isogenic disease line MEA in lighter blue. You can already see from these plots that the bursting activity was very different. So comparing the two lines by their burst duration, the disease line was characterized by a longer burst duration, as it's evident also from above, a longer interburst interval. And instead, the two lines were very similar in the interburst interval coefficient of variation, so very similar bursting regularity. We also compared the two dopaminergic neuron lines, the wild-type line and the isogenic disease line. In this case, the wild-type line in red had a longer burst duration and also a longer interburst interval. Instead, the disease line in orange had a higher interburst interval coefficient of variation, which means that this bursting pattern was more irregular. So what we realized is that these four cell lines that we analyzed had a very peculiar and characteristic bursting. So what we did was to observe and analyze this network shape. So to do so, you see here represented human motor neurons, the wild-type line in blue, in one MEA at the in vitro 14, and in another, and the same one recorded at the in vitro 21. And then in light blue, you see another MEA, this time with diseased motor neurons cultivated on top, cultured on top. And you see that the recordings at the in vitro 14 and the in vitro 21. So to compare the shapes of these two cell lines, we created some templates. These templates were created by averaging each recorded burst in one MEA and across MEAs for a specific in vitro and cell line. So in total, we obtain 4 templates for the human motor neurons and for the human dopaminergic neurons. What you see here on the right side of the slide is how we could differentiate the 2 cell lines. For example, let's observe the first template, the wild-type human motor neuron template at the in vitro 14. We perform with this template the linear correlation with all the wild-type MEAs and all the diseased MEAs. With the linear correlation with the wild-type MEAs, as expected, we found a Pearson coefficient similar to 1, which showed the robustness of the template that we created. While performing the linear correlation with the diseased MEAs, we found a lower Pearson coefficient, and so we could differentiate between the 2 cell lines. We repeated the same kind of analysis for the human dopaminergic neurons. This time was a bit more complicated because, as you can see for the in vitro 14, the 2 lines had a very similar oscillatory pattern. However, taking, for example, in the right side of the slide, the wild-type template at the in vitro 14, and performing the linear correlation with all the wild-type MEAs and the diseased MEAs, even if the Pearson coefficient was very similar to 1 in both cases, it was still possible to find differences between the 2 cell lines. And this because the bursting was really characteristic of the cell lines. Until now, I showed you a bit broader metrics, so macroscopic metrics that concern the network or single cell. And now I would like to go to subcellular resolution. That's really a strength of the high-density microelectrode-arrays. What you see on the left side of the slide is what we call a neuron footprint, so a single neuron action potential spatial distribution. Each line represents 1 electrode recorded for a certain period of time. When you see here in red instead, with the red waveform, the starting point of propagation of an action potential that propagates along the axonal direction, that is here represented in dark blue in the first plotting and in light blue in the second one. By alignment, temporal and spatial alignment of all the detected action potentials, we could compute the spike propagation velocities. Now, looking at the right side, at first, we wanted to see if the velocities changed in the wild-type motor neurons. And what we noticed is that there was a decrease in velocity between the in vitro 28 and the in vitro 42. For what concerns the disease line, instead, there was an increase in velocity between the in vitro 14 and 28. And the 2 lines differed just at the in vitro 42. Now, the last application that I would like to show you today is a very important application for high-density microelectrode-arrays, and it summarizes all the metrics that I showed you up to now, which is the computation and evaluation of the effect of a compound on the activity of the neurons. In particular, we did it in motor neurons, and we used as a drug retigabine, which is an anticonvulsant drug, a Kv7 potassium channel opener, and it's currently used to contrast the hyperexcitability of motor neurons in the SOD1 mutation of amyotrophic lateral sclerosis. So what we would expect is a decrease in the firing rate. So what you see here, it's 4 different MEAs. So the map that I showed you before is the firing rate recorded in an untreated condition. In the second line, you can see the same 4 MEAs, this time recorded when administering the vehicle control and 1, 5, and 10 micromolar of retigabine. You can appreciate how the overall activity and the firing rate decreases with the increased retigabine concentrations. On the right side, you can also see how the percentage of active electrode decreased, and how it was possible to differentiate between the vehicle control and each retigabine concentration, 1, 5, and 10 micromolar. Also, looking at the network bursts, you see here represented the 4 MEAs as before in an untreated condition, and then when administering the vehicle control, and 1, 5, and 10 micromolar of retigabine. In this time, for example, with the 5 micromolar, we observed a decrease in the frequency of this oscillation, and at 10 micromolar, they even disappeared. Last, looking at we wanted to analyze the number of spikes per minute, and we used 3 different configurations. The first one is a configuration which comprises 1,020 electrodes placed in the active regions. The active regions are the ones characterized by the highest firing rate. When using this kind of configuration, it was possible to find differences in the number of spikes per minute between the vehicle control and 1, 5, and 10 micromolar concentration, so all of them. And moreover, the data dispersion was very little. Then we use a second configuration of 8 × 8 electrodes in a grid placed every 200 micrometers. In this case, it was possible to find differences between the vehicle control and 5 and 10 micromolar of retigabine, and not between 1 micromolar of retigabine and the vehicle control. Moreover, the standard deviation increased. The last configuration is a 4x4 electrode grid of electrodes placed every 200 micrometers again. And in this case, it was not possible to find differences between the vehicle control and all the applied retigabine concentrations. The standard deviation increased even more than in the previous case. So the message that I would like to give you with this slide is that it's very important to place the recording electrodes in the regions where neurons really are. And also it's important to have the availability of multiple recording sites. So the more the electrodes, the better the results. And with this, I'm already at the conclusions of my presentation today. Today, I showed you how we could study the electrical phenotype characterization of different cell lines. We could also propose the action potential propagation velocity and the network bus shape as potential new biomarkers. And we could also obtain very high reproducibility in drug effect evaluation. As an outlook, if you remember at the beginning, I told you that we use 83 MEAs. So a platform like the MaxWell would offer high throughput screening and allow to test multiple compounds and more drug concentrations. We could also try to answer different scientific questions, for example, evaluate the differentiation stage or try to recognize a specific cell type. And we could also work towards an automatic classification of the cell types using some metrics that are really characteristic of the cell type. And today I showed you the network shape as one of them. With this, I would like to thank my group, my Professor Andreas Irleman, Michele Fischel, Alessio Buccino, Manuel Schroeder, Sridhar Kumar, and Gustavo Prak. These are all co-authors of the paper that we recently uploaded in bioRxiv. And I would like to thank the two funding agencies, ERC and InnoSwiss, MaxWell Biosystems for the scientific and technical support, FUJIFILM Cellular Dynamics, and you all for your attention. Thank you very much.
Thanks a lot, Silvia. That's very nice, and thanks as well, Simon. So we are very excited to see all of these results combining MaxWell Biosystems and FUJIFILM Cellular Dynamics cells. And now let's go to the question and answer portion. We are getting a lot of questions. Thank you so much. So please continue to add your questions in the chat. So the first question is for Simon. Yes. So, for regenerative medicine, we have a question here asking, when could the iPS cells from cellular dynamics be used for patients?
Oh, sure. So, yeah, to be sure, all the stuff that we're talking about today, these are our cells that we're producing just for research use only. So just for, looking at basic science and drug discovery efforts. CDI does have some interest in and some programs looking at cellular therapy, but hopefully soon. But I can't comment on timelines for that.
Okay, thanks a lot. Yep. Okay, next question. This is actually for Silvia. Silvia, if you can come up.
Here I am.
Yes, thank you. So the question here is about your recording protocol from day 14 to day 21.
Yes.
Are the number of electrodes the same, or are you comparing activity only from the active electrodes?
For the network, or?
I think you can mention for all of the assays.
So when we do these kind of recordings, first we scan the whole electrode area. So we go with configurations of more or less 1,000 electrodes covering all the electrode array. And then when we want to perform network recordings, we really want to target the most active regions and perform simultaneous recordings. And we have the availability just of using 1,020, but of using 1,020 at the same time. Then we select the most active electrodes each time. So the electrodes would change, not when administering a compound. In that case, it's exactly the same electrodes, pre and post treatment.
Okay, thank you very much. Next, still for Silvia. Yes, but I think also Simon can comment later after Silvia. So this is about the cells. Mm-hmm. So did you use a cell line coming from only one healthy donor, or did you compare lines coming from different healthy donors? If you did comparison, so maybe the first question, like are the wild type cells coming from one donor or from multiple donors?
So they come from the same, right? So maybe Simon can comment more on this. They come from
Sure. Yeah, so I think all of the lines Silvia was talking about today, these all came from a single donor, and so there's multiple ways you can do this. But in the case of what Silvia talked about, the healthy controls were all from one donor, and then the patients were still the same donor but just engineered mutations. So.
Okay, thanks a lot. And maybe Simon, if you have insights, if in case we are comparing the activity of same wild type, but different donors, do you think there would be a difference between the results?
Yeah, I mean, it'll depend, of course, on the particulars. Obviously, genetics is going to play a role, but I think in these cases, some of those results were really striking, those differences, and I think it's that mutation difference was, I think, would be likely to hold true across backgrounds.
Cool. Thank you so much. Okay, now back to Silvia. So about the drug application,
Yes.
At what day did you treat the cultures with retigabine?
So we treated the culture in this kind of experiment at the in-vitro 14, where the network was really connected, and we could nicely evaluate across different MEAs. However, we also tried to perform other recordings at the in-vitro 21. But yeah, they are not in the presentation today. But yeah, the in-vitro 14, following also other protocols that were online, even if it was not exactly the same cell lines. But now we are working on comparing also different the in-vitro.
Oh, wow. So stay tuned for the next publication, I would say. Follow up on that. Were you able to wash out the drug effect and bring back the firing levels back to normal?
So actually, we didn't try to wash. Actually, we kept it on the MEA, and we recorded also again after a couple of days in vitro to see if the drug still had an effect, and it was still really shutting down kind of the firing rate of the cells. But we didn't try to wash it, no.
Okay, yeah. Maybe some more ideas for next experiments.
Totally.
Yes.
Yeah.
Cool. Again, for Silvia, did you compare the disease model ALS with normal cells in single cell state for the axonal action potential propagation? Because I think maybe this question. So just for the axon results, did you also do it for ALS?
So yes, it was in the graph. So
maybe if you can share that.
Yes, of course. Let me go to the slide. And before here. Yes, so this one was also for the ALS, and it's actually represented in the line below. You can see here one neuron of the ALS line, and then the alignment of the action potentials in space and time, and then the computation with the linear regression of the spike propagation velocity, and then the comparison of the velocity over time, and then and all these are all single neurons, so there is spike sorting that it's done to really, I don't know if you're all familiar with it, but really to be sure that this is just one neuron and it doesn't come from the overlap of multiple neurons.
Yeah, thanks. Okay, we have a nice comment here for you, Silvia. Yes, saying nice presentation, Silvia. And of course, a follow-up question. Did you try synaptic events recording?
No, we didn't try this.
Okay. Yep. Next, this is now about MEAs. So what materials are there in the MEAs? So when you plate or culture the neurons, it's very similar to what you would do. Sorry, maybe can you see my screen or?
No.
Okay. Maybe I'll just.
Because I.
Sorry about that. Or okay. Anyway, so can you also see the videos or not? No, okay. I guess that's fine. I'll just continue with the questions. So about our MEAs. So the electrodes are platinum, and we have platinum black to make the impedance lower, improve the signal quality basically, and the surface itself is just like glass. So the same protocol that you would use for glass petri dishes for plating your cells or glass plates would work as well on the MEAs. Okay. Yes. So next question. How do you determine the ratio of astrocytes to neurons for the co-culture? So I think this is for Silvia.
Sorry?
How do you determine the ratio of astrocytes to neurons?
We actually dilute the vials that we receive. So we know that we get 1 million astrocyte vial, and then we dilute it with the neurons. And for the neurons, we do the counting with the trip-on blue and the mass spectrometer, and then we dilute them in the right ratio.
Mm-hmm. Okay, thank you. Is there a difference in network activity if you change the ratio of astrocytes and neurons?
I think it's to be expected. I don't know exactly if it has an effect on the oscillations. I guess they would still be connected. I know that from some publication they show that the different ratio of the astrocytes can affect, for example, the overall firing rate of the culture. Anyways, astrocytes are used to sustain the neurons, so I guess they bring in some positive effect on the neuronal cultures, and I guess the different ratio could affect, for sure, the firing rate.
Yeah. Okay. Next, I think this is for both Silvia and Simon. Do you have any comments if there are any molecular changes or plasticity that contribute to changes in MEA network activity in between, across multiple days and also between cell lines?
So this
That's a tough one.
Analyze this effect.
So I guess, for multiple days, what I can say is that the network is just getting more matured. That's why you see more bursting activity as you record much longer. Not comparing between cell types, understanding the mechanism that is involved in these phenotypes, I guess, is something for further studies.
Yeah. Basically, the networks take time to develop, and they kind of build on themselves. The neurons that wire together, they kind of build up. So it takes a little bit of time. So they got to form the synapses, form those networks and stuff. So I think that's the primary thing that you see. But takes a while.
Mm-hmm. Cool. Okay. We have more questions. I think we can have just 2 more. Next is, do you expect the performance and viability of neurons in presence of astrocytes will be improved compared to neurons alone?
Yes, I would expect this. Definitely.
Mm-hmm.
So the presence of the neuron, of activity of the neurons, and the neurons need astrocytes, right? And the presence of the astrocytes would for sure also increase the survival of the neurons and the MEAs.
Mm-hmm. Yep.
Yeah, it depends a little bit on what question you're trying to answer as well. Sometimes it's helpful to have the monoculture to really isolate your variables, but in general, the astrocytes do seem to make a lot of things nicer with looking at MEA and things like this. The glutamate uptake really helps with cleaning up the networks and getting a more synchronous network.
Yeah. And maybe as a follow-up question to that, is there actually an effect of astrocytes that the network activity is much earlier as compared to just neurons alone?
This we didn't try,
Mm-hmm. Okay.
so I wouldn't know how to answer,
Mm-hmm.
but it's a very interesting question. This I really don't know.
Okay, yeah, but I think this is one of the questions that can also be answered using
Yes.
the technique.
Of course.
Yep. Yeah, thank you so much. Thanks again, Simon and Silvia. So these are some information about MaxWell Biosystems and also how you can contact us for additional questions that you have. You can send us an email in info@mxwbio.com. You can schedule a call through Calendly, or you can visit our website. And once again, I'd like to point you to our files. You can download the protocol of plating FCDI cells on MaxOne chips by downloading that and checking it. I think Silvia also did the same protocol for experiments. And you can also download Silvia's paper in bioRxiv as in the link here. Okay, thank you very much. So you can meet us in Bio Japan. We have a booth there, a virtual presentation tomorrow, and we also have a partnering program. And we're also here in a very local event, our TED annual meeting here in Switzerland. Thank you very much. So for those questions that you're not able to answer, please don't worry. We will send you an email to answer these questions. Thank you very much, and have a good morning, evening, or noon, wherever you are in the world. Thank you very much.
Thank you.
Bye.
Thank you so much. Bye.